So, as I just summarized, direct current stimulation can change synaptic efficacy in the brain. And if you can change synaptic efficacy, you may be able to have profound effects on cognitive function, on behavior, and on therapy as well. I wanted to remind you that we shouldn't just be thinking about the soma anymore, though we have conducted recordings from the soma and found that the soma of cells changes in response to stimulation. The dendrites are also always stimulated, and the dendrites are very important for the processing of synaptic input. And the axons, so the inputs themselves, are in fact the most sensitive structures in the brain, and they're critically involved in synaptic processing. And so again, when we apply direct current stimulation to the brain, we polarize the soma, the dendrites, and the axons, and all of these together produce a cumulative effect in how these cortical columns are processing activity. And terminals, again, playing a very, what we believe, are an important role. And one of the reasons is that there are really very many terminals in every cortical region, and they're pointing in every possible way. And the reason I say that is it basically means that no matter how direct current stimulation is passing through the brain, we're going to be polarizing these nerve terminals, these axon terminals. And as I mentioned before, while the outcomes can be complex, they're very deterministic. So we have very precise now neurophysiological models that allow us to predict when you apply direct current stimulation to a particular cortical column that's processing information in a very particular way, what happens. We really just need three pieces of information. One is the direction of the current flow relative to the column. Is it in? Is it out? Is it sideways? We need to know which membrane compartments are being polarized by the stimulation in that case, in which pathways are being activated. And with these three bits of information, we can actually predict with a lot of precision how synaptic processing is changed by a cortical column. Now, I need to finish by bringing us all the way back to functional selectivity, which is the point I started with in the second part of this talk. And I want to show you just a couple more experiments. This is an experiment not from my lab, but from the lab of Leo Cohen at the National Institute of Health, but with a brain slice as well. And the short version of what they did is they used a brain slice and they gave it a very weak stimulus. They just gave it a weak synaptic activation that was not in itself enough to produce plasticity. So basically they had weak ongoing activity that did not produce synaptic plasticity. Now they combine that weak ongoing activity with tDCS. And what they found was now they could produce plasticity. What that means is if the brain is active in such a way that it's not able to produce learning, when you apply tDCS at the same time, you can now change it into a mode where it can. And so what you're seeing here is the same measures I showed you before, these EPSPs, these measures of synaptic efficacy. Here's the baseline activity, it's going on and nothing's happening. All of a sudden direct current stimulation is added and you produce a change that not only happens during tDCS, it keeps growing, it plateaus, and it can last 2 hours after the tDCS as well. So the tDCS initiated what's called long-term potentiation, a strength in synaptic connection, and this is analogous to learning. The experiments we've been doing in my lab are a little bit different in that we've been looking at the effects of tDCS when you already have plasticity. So in this case, we're using a phenomenon called theta burst stimulation. Theta burst stimulation is something that can be done both in animals and in humans, where intense stimulation is applied to the brain in bursts like tsk, 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 and that intense stimulation can produce plasticity. And this is not necessarily with tDCS. And so what you're seeing here in the black trace is baseline synaptic strength, theta burst activity applied without tDCS, and we see an increase in the strength of these synapses. So what that means is that theta burst stimulation produced a strengthening of synapses. This again is what's called long-term potentiation, and the strengthening of synaptic efficacy that lasts a long time after the intervention itself, in this case over an hour, is believed to be associated with learning. Now, we also conducted experiments where we did theta burst stimulation at the same time as the application of direct current stimulation. And that's what you're seeing here in the pink trace. So we applied now theta burst plus tDCS, and what we found was that the same theta burst stimulation in combination with tDCS produced a bigger effect, and that effect lasted a long time. The direct current stimulation did not produce plasticity. If you apply direct current stimulation on its own, you don't see any effect at all. What produced the change was the theta burst. It was this ongoing plasticity that was already happening. And what tDCS did was it made it more effective. Now, the reason this result is important, and that's what is shown in this experiment here, is that you can get theta burst in one group of synapses, but not in others. Meaning, if one part of the brain or one set of synapses is learning and getting stronger, other parts of the synapse may not be. And if you apply direct current stimulation in this situation, you will enhance the ones that are learning, you will boost the ongoing synaptic plasticity, but you won't affect the ones that are not learning, because we and Fritsch and others have shown that when things are inactive and you apply tDCS, you do not produce and you cannot modulate synaptic plasticity. Direct current stimulation changes ongoing synaptic activity. It changes ongoing synaptic plasticity, and where you do not have activity or where you do not have plasticity, tDCS is ineffective. And this is actually functional selectivity. This means that if you're thinking something, if you're learning something, and a particular set of networks in your brain become active, tDCS can affect them. But other networks that are inactive will be unaffected. And I just want to show you one more result, which is from my lab, which we find very exciting. What you're seeing here is an experiment where theta burst, let's look first at black, theta burst on its own, is applied again and again and again. So every time you see a line, it's applied again and again. So you can see that this theta burst, this endogenous sort of synaptic learning, produces a gain. And if you do it theta burst again, you can get another gain. But as you kept doing it more and more, you sort of plateau. You can't get, in this case, more than a 40% increase in strength during normal theta burst. Now, in the pink trace again, we're applying direct current stimulation. So here's direct current stimulation applied once, and you can see that you can get a little bit more and it lasts longer. Here we do theta burst plus tDCS again, and we get more enhancement. And that enhancement is maintained. What that means is that tDCS doesn't just make us learn faster, it's able to get us to a higher maximum level of learning. Now, I'm using learning here in a very abstract sense. What I'm only showing you here directly is synaptic efficacy and the fact that tDCS can boost both the rate and peak of long-term potentiation, so this long-term strength in synapsing. But there are many people who believe strongly that this is an analog for learning in people. So in the interest of time, I'm going to only very briefly discuss tDCS and active network. So as I mentioned before, the brain is not inactive, it's always active. And sometimes this activity manifests itself in ongoing oscillations. So these are organized activity in the brain. People will call it alpha oscillations, gamma oscillations. When you sleep, people call it slow wave oscillations. And these oscillations are considered absolutely and profoundly critical both for normal brain function and they've been linked to pathological brain function. And we and others have shown that direct current stimulation influences these oscillations. So this is just an example of one experiment from my group where we applied direct current stimulation to brain slices that were ongoing gamma oscillations. This is gamma oscillation recorded extracellularly in a brain slice. And we also apply this in a network model to be able to test some hypotheses. What we found, so this is baseline gamma power. This we're showing here percent change, so initially it's stable. We found that when we applied direct current stimulation, the nodal direction would produce more gamma oscillations, so the gamma power got bigger, while the cathodal stimulation will produce less oscillation. And when we turned off stimulation, we saw this interesting rebound effect, and then it went back to baseline. What's important to understand here, a nodal tDCS did not produce gamma oscillations. These gamma oscillations are already present in the brain slice, or they can be produced in the brain slice by different perturbations like adding carbachol. The slice is generating oscillations, or more generally, the brain generates oscillations. And the origin of oscillations are very complex. They involve a loop between excitatory and inhibitory cells often, and very specific network architecture. And nodal tDCS, the 9-volt battery, cannot produce oscillations. Oscillations are much too complicated. But because a nodal tDCS polarizes cells, and because polarization affects oscillations, when we apply nodal tDCS, we can change ongoing oscillations. So a 9-volt battery can't produce oscillations, but a 9-volt battery can change oscillations. And we've tested this on a variety of different systems, as have other people, and it turns out that universally, brain oscillations are very sensitive to weak current. Now, if these brain oscillations are involved in learning or dysfunction, then they are an avenue to get to learning and to get to treatment. tDCS changes the oscillations, and then the oscillations play a role in learning or behavioral treatment. And so here again, a power modulation produced by tDCS, but tDCS did not generate oscillations on its own. And these acute adaptation effects end up being very important for linking these effects of oscillations with plasticity. But I'll simply refer you to our papers now and just quickly summarize by saying we have plenty of data showing that when the brain is oscillating and you apply a weak direct current stimulation, you can change the nature of that oscillation. And if you change the nature of the oscillation, you can change how this cortical column processes input. So it's yet another way where we can think about how direct current stimulation can change how the brain works, not by generating activity, but by changing ongoing activity. And we've considered this in exhaustive detail, not just for DC, but for other stimulation frequencies as well. And I'll refer you to the paper to do that. And I'll also refer you to another study where we've actually linked this stimulation with changes in slow oscillations with results that have claimed that tDCS can actually accelerate sleep homeostasis as a way that might actually enhance memory consolidation during sleep. We didn't do the experiments themselves that were conducted in human. These were conducted by Lisa Marshall and were published in Nature. But what we were able to show is that these computational models that include cell polarization and oscillations can provide a link from brain stimulation to learning. So to summarize, what do we know about direct current stimulation and oscillations? We know that they are sensitive, not just gamma, but actually every oscillation that has been tested is very sensitive to direct current stimulation. Why it's sensitive is not always a simple answer. It relates to the complexity of the oscillations themselves, but we know it's related to the polarization of cells. And it turns out that these active networks become extra sensitive to direct current stimulation because they're oscillating, because they're so finely tuned. We discovered that active networks are very sensitive to direct current stimulation. And in another paper, we were able to show that if you sustain this modulation of oscillations with direct current stimulation, then it itself can lead to plasticity. So here's the big picture summary. tDCS produces weak current flow in the brain. This is not a problem with tDCS. This is not a secret that no one wants to talk about with tDCS. It is very well known for decades that direct current stimulation produces weak current in the brain, and our own contribution has been to develop these very detailed computational models that I've been showing you how current flow flows through the brain. This weak current flow will produce a biphasic membrane polarization. So there's no such thing as a depolarizing tDCS. Even a nodal is not simply depolarizing. A nodal tDCS is depolarizing of the soma. How much? Turns out not very much. The soma is less than a millivolt. Axon terminals maybe just over a millivolt. But just because you polarize a little bit doesn't mean you can ignore the effects of tDCS, because there's many situations, many ongoing processes in the brain where a little bit of polarization can produce a big effect. And one of those places in which we talk about synaptic efficacy and relate to that synaptic plasticity. Synaptic efficacy and synaptic plasticity are known to be sensitive to little changes in membrane polarization. Little changes in membrane polarization will change the amount of plasticity that you achieve. And what's really nice about this is that it's pathway specific. So a pathway that is ongoing plasticity versus a pathway that is not, only the pathway that is ongoing plasticity will receive the benefit of direct current. And we can expand on this through very detailed work looking at oscillations and direct current stimulation. I want to conclude by saying, therefore, that I've spent the second part of the talk going from anatomical specificity to functional specificity. And just like with anatomical specificity, where we have extensive data and extensive modeling to inform what we're doing, it's the same thing with functional specificity. And so while we certainly haven't answered all the questions that exist with tDCS, I would say that compared to many other interventions, including most brain stimulation interventions, and certainly pharmacological interventions, we understand much more about how tDCS works, how it might affect disorders like tinnitus and other neuropsychological disorders. And we have a rational basis for optimization. And what I'm hoping is that the information I've shown you here, which was obviously not clinical, it was engineering and basic science and physiology and nature. What I'm hoping is that some of the insights I've shown you here may actually be able to inform ongoing clinical trials, hopefully to make them more rational and ultimately to make them more effective. So thank you for your attention.